Hello everyone, am I live? Can you see me? Can you hear me? Kindly give me confirmation if I'm clearly visible, audible. Let me confirm. If I'm clearly visible, audible, I will start the class ahead. I hope, yes, it's working and I'm clearly visible and audible. So I welcome you all for today's session. A very, very good evening to all of you. I am Dr. Priyanka Sachdev here, and today I am here to continue my series that is bacteriology. So this series is basically for second prof MBBS students for microbiology who are appearing in their university exam this year. So many students requested me to launch this series because they are finding bacteriology very difficult. So I found out a unique way of studying bacteriology. Today morning, we have already started bacteriology and in a unique way. What is unique in the series? So I'm going to teach you all 40 bacteria in your syllabus one by one in a comparative manner. So first, we have started with gram-positive cocci. In gram-positive cocci, in the morning, I taught you three bacteria in the morning, right? So I started with Staph aureus, then I moved on Streptococcus pyogenes, and then I move on Streptococcus pneumoniae, that is pneumococcus. So these are three gram-positive cocci. We have covered them in a beautiful comparative manner for the better retention in episode 1 of bacteriology. Those who have missed it, they must watch the recording. It is available on the YouTube Complete Bacteriology episode 1. So always I'm having a fixed protocol in which I teach the bacteriology under the following headings only. So for all bacteria, we cover bacteriology like this only. So with the introduction, I always tell you the name of the scientist who has discovered that particular bacteria. So sometimes one-liner questions, MCQs comes on the scientist. So for all the bacteria, at least you must write the name in a comparative manner. So when any MCQ come, based on the scientist, it may click in your mind that this is the correct answer for this particular bacteria, right? Then you must know the classification of that particular bacteria. It is must. Without that, you cannot pass your second prof exams, right? In morphology, always tell three things: whether capsule present or absent, capsulated, non-capsulated, whether spore present or absent, and whether motility present or absent. So always comment on these three things, and I have told you beautiful mnemonics to find out whether the bacteria are capsulated, non-capsulated, spore forming, non-spore forming, and motile and non-motile. After that, in culture, just enumerate the various culture: blood agar, nutrient agar, whatever, McConkey agar, and write the special one lines in them. Right. In biochemical reaction, only write the important biochemical reaction, enumerate them, and write principle, method, interpretation. For all biochemical reaction, you have to write like this only in your exam. So introduction, the principle, interpretation, and method, right? Antigenic structure not really important. In some of them it is important. Resistance not important. Virulence factor are must. You must know the virulence factor of all the bacteria. Pathogenesis may you must enumerate the name of the diseases they are causing. Nowadays the pattern is changed. In many universities the bacteriology is like system wise. It is not bacteria wise. For that, you must know the diseases for each bacteria. Still, I'm teaching you bacteriology bacteria wise only because it is easy to retain the bacteria in bacteria wise manner. Now you see the diseases of all. Just suppose Staph aureus causes skin diseases and GIT diseases, that is food poisoning. Streptococcus pyogenes causes cardiac disease, that is rheumatic carditis or rheumatic heart disease. It causes post streptococcal glomerulonephritis, that is renal disease, and it also causes skin disease. It also causes food poisoning. So it is coming in four systems. It is coming in GIT because it causes food poisoning. It is coming in CVS because it causes carditis. It is coming in renal system because it causes glomerulonephritis, and it is coming in cutaneous system, the skin, also because it causes various pus forming lesions in the skin and respiratory tract, right? So in this way you are going system wise also. System wise we can cover bacteriology in both manner, the bacteria wise, the system wise, right? So in the morning we have already covered gram positive cocci. We have already covered gram negative cocci. Gram negative cocci in the morning in episode 2 of bacteriology. I taught you the two Neisseria. Neisseria meningitidis, that is meningococcus, and Neisseria gonorrhoeae, that is gonococcus. So we have covered two more coccus, meningococcus, gonococcus. Both of them are gram negative cocci. Basically in the morning we have covered gram positive cocci and we have covered gram negative cocci. Now it's time to start bacilli. Let's start bacilli. Let's start bacilli. In the bacilli, first I will teach you. Till now we have covered gram positive cocci and gram negative cocci. In gram positive cocci, mainly I taught you two bacteria: Staph aureus, Streptococcus pyogenes, and Streptococcus pneumoniae, that is pneumococcus. In gram-negative cocci, I taught you two bacteria, Neisseria meningitidis, that is meningococcus, and Neisseria gonorrhoeae, that is gonococcus. Now, I am starting gram-positive bacilli. And in the end, I will move on gram-negative bacilli. That is the biggest category. In gram-positive bacilli, I will start with Corynebacterium right now. So, right now, I am starting with Corynebacterium diphtheria. After that, I will move on Bacillus anthrax, the second bacteria. After that, I will move on Clostridium. So these three gram-positive bacilli, I am going to teach you one by one right now in this lecture. After that, I will move on gram-negative bacilli, in which I will teach you E. coli, Klebsiella, Proteus, Salmonella, Shigella, and Vibrio. So these are the important gram-negative bacilli that I will take in episode number 4. So episode number 3, basically this episode which you are watching currently, I am going to teach you these 3 bacteria, and if time permits, we will start with gram-negative bacilli also. So in this episode, we are starting with gram-positive bacilli. Give me a thumbs up. Shall I start? Are you there? Kindly someone give me a thumbs up in the chat box. I can see your chat. Let me start with Corynebacterium diphtheria. So let me start with Corynebacterium diphtheria and Bacillus anthrax together in a comparative manner. So please, all the students, please do this comparative table with me. Make this comparative table with me. That is Corynebacterium diphtheria and Bacillus anthrax. Please do in a comparative manner like this. Let me start with Corynebacterium diphtheria, right? So let's start with Corny. So first we will fill this, and then in comparative manner we will fill this, and then after that I will start with Clostridium. There are 4 Clostridium I am going to teach you one by one. Clostridium perfringens, Clostridium butyricum, Clostridium difficile, and Clostridium tetani. All of them are important. So 4 columns more you have to make here. So total gram-positive bacilli will be done. 1 Corynebacterium diphtheria, 1 Bacillus anthrax, and 4 Clostridium. So total 6 bacteria in a row you have to do. Give me a thumbs up. Shall I start? Yes, Dr. Mahesh, you can see, I guess. So let me start. Let me start with Corynebacterium diphtheria. Write down the introduction. In the introduction, write down the name of the scientist, important scientist, which have contributed in the discovery of Corynebacterium diphtheria. Write down 4 scientists here. If you wish, you can write down all 4. The most important is the name of the scientist is Bretonneau. Bretonneau is the name of the scientist who has discovered the disease diphtheria. It is caused by a organism which is forming a pseudomembrane. So the first discovery was done by Bretonneau, right? The second, the actual bacteria was discovered by Clapp and Loeffler. Clapp and Loeffler. That's why the name of the bacteria is Clapp-Loeffler bacilli. It is an important MCQ. Clapp's Loeffler bacilli is the another name given to Corynebacterium diphtheria. Corynebacterium diphtheria का दूसरा नाम है Claps-Loeffler bacilli. Give me a thumbs up. The exotoxin of the bacteria Corynebacterium diphtheria is discovered by Raux and Yersin, right? And the antitoxin, this is toxin. Antitoxin is discovered by von Behring, who is given Nobel Prize for that. So learn the name of the 4 scientists who discovered the disease diphtheria, who discovered the bacilli, who discovered the toxin, and who discovered the antitoxin. Please write down the name of the scientists. Frankly speaking, I don't remember the name of all scientists here, but the most important is Clapp and Loeffler, who has discovered the bacilli. That's why the name of the bacilli is Clapp-Loeffler bacilli. But at your stage, you must know all scientists here. Who has discovered the disease? Bretonneau. Who has discovered the toxin and who has discovered the antitoxin? For toxin, Raux and Yersin, and for antitoxin, von Behring. These are the name of the 4 scientists. In the introduction, please write down the name of the 4 scientists. The one who discovered disease, the one who discovered the bacteria, the one who discovered the toxin, the one who discovered the antitoxin. Write down 1, 2, 3, 4. MCQ can come on any point. You never know. You never know. Coming on the classification. Everyone give me a thumbs up. So in the introduction, we have written the name of the 4 scientists. The one scientist who has discovered the disease diphtheria, the second scientist who has discovered the bacteria, bacilli, diphtheria, Corynebacterium diphtheria, bacilli को किसने discover किया है? Clapp and Loeffler. That's why it is known as Clapp-Loeffler bacilli. The one who has discovered toxin, the one who has discovered antitoxin. जिसने antitoxin discover किया है उसको Nobel Prize मिला है. Nobel Prize is given to only one of them. That is the one who has discovered antitoxin. That is von Behring. In 1890, he got the Nobel Prize for that. So learn the name of the 4 scientists in front of you. 1, 2, 3, 4. Give me a thumbs up. Easy till now. Coming on classification of Corynebacterium diphtheria. So this is okay. Classification, I am coming on it a little bit later on. First, let me teach you the morphology. In morphology, tell me whether it is gram-positive and gram-negative. It is gram-positive. Whether it is cocci or bacilli, it is bacilli. So they are gram-positive bacilli. Tell me capsule present or absent. Tell me spore present or absent, and tell me motility present or absent. Comment on the three things. Would you like to comment? Anyone, Dr. Mahesh, others? I can't see other name. Can you comment on the three things? Capsule, spore, and motility. Is it present or absent in Corynebacterium diphtheria? To comment on the three things, I have given you three mnemonics. Apply the three mnemonics. See. So it is a gram-positive bacilli. It is gram-positive and it is not cocci. It is bacilli. What is the shape? The shape is rod shape. What is the color? The color is green. The color is green. The bacteria is green color. Can you see green color rods? Yes. You all can see green color rods. It is green color on Albert stain. This stain which is shown in the diagram, it is Albert stain. It is not stained by Gram, usually Gram stain. It is stained by Albert stain. And on Albert stain, the bacteria is green color. And on the poles, on the two poles, the purple or blue color granules are present. And bacilli are not present alone. Either they are V-shaped or they are L-shaped or they are K-shaped. V, L or K. So they are present like this. So because this is one bacteria, this is second bacteria. They are not separated completely while doing binary fission. So this is one bacteria. The bacteria is doing the binary fission. While binary fission, the one bacteria converted to two bacteria, the two daughter cells are formed. But they are not completely separated. So that's why they are V-shaped, L-shaped, K-shaped. That is the reason. And they are green color. They are basically green color. Let me form them with green color. This is like green color, like this. At the periphery, at the poles, they are having purple color granules. These granules are known as metachromatic granules. Give me a thumbs up. These granules are known as metachromatic. Now it is looking like a Chinese language. That's why it is known as Chinese letter arrangement. Chinese letter arrangement. This arrangement, it is a very important MCQ and one-liner question in university exam also comes. Chinese letter arrangement seen in Corynebacterium diphtheria. The reason is that two bacilli occur together in the form of the V, L and K. That's why it is looking like a Chinese language. I don't know Chinese language, but yeah, it looks like this. So Chinese letter arrangement, because during binary fission, the two bacilli are not separated from each other completely. They remain stick with each other. That's why they are V-shaped, L-shaped or K-shaped, looking like Chinese letter. Also known as cuneiform arrangement. This arrangement also known as cuneiform arrangement. Tell me the color coding. The bacteria is green color. Can you see the bacteria is green color? And the metachromatic granules are of blue or purple color. The meaning of the metachromatic is blue color. Metachromatic is blue or purple color. Since the metachromatic granules are present at the poles of the bacteria, these are known as polar granules. These are also known as polar granules. So let me draw a bacteria. The bacteria is green color like this. Two bacilli are stick together. That's why V, K or L shape. And it is having a metachromatic granules at the poles. So what are the names of the metachromatic granules? It is known as either metachromatic granules or known as polar granules. The meaning of the polar, because they are present at poles, North Pole, South Pole, polar granules. It is discovered by a scientist known as Babes Ernst. What a good name. Babes Ernst. Babes Ernst. That's why known as Babes Ernst granules. And the first bacteria in which these granules were discovered is not Corynebacterium diphtheria. It is Spirillum volutans. That's why known as volutin granules. So these are the four names given to these granules. The reason for each of them you should know. Why they are known as volutin granules? Because it is discovered in a bacteria, in the volutin bacteria. It is discovered by a scientist Babes Ernst. That's why known as Babes Ernst granules. It is blue color, purple color. That's why metachromatic granules. And they are present at the poles. That's why polar granules. So these are synonyms of each other. Give me a thumbs up if you got it. On the morphology, draw this diagram. Say these are gram-positive bacilli. These are pleomorphic. You can see these are green color bacteria. Albert stain with the bacteria is green color and purple color granules. They are present in angular fashion, either V or L. It is looking like Chinese letter or cuneiform arrangement. It is due to incomplete separation of the daughter cells during binary fission. I taught you everything, right? They are club shaped. They have metachromatic granules at the end. Metachromatic granules also known as polar bodies because they are present at the poles, and they are also known as volutin granules and Babes Ernst granules. These are the four names given to the granules, the purple granules which are present at the two ends. You should know the four names. These are synonyms of each other. Everyone give me a thumbs up. Either volutin granules, Babes Ernst granules, polar granules or metachromatic granules. Everyone give me a thumbs up. You can see it here, right? Now, why these granules are present? Basically, what is these granules? Let me draw a bacteria. Let me draw the granules. Actually, these granules contain stored phosphate. They contain phosphate inside them. So they contain stored phosphate inside them. Whenever any bacteria divide, phosphate is required for nucleus. for nuclear division. For nucleus, it is required. But whenever any bacteria is not dividing, so phosphate get accumulated, and it is accumulated in the form of the granules. So basically, these granules are formed when the bacteria is unable to divide in starvation. Like in the condition starvation is there, no food is available, so bacteria can't divide. If the bacteria can't divide, nucleus is not dividing. Nucleus is not dividing, so phosphate is not required. Phosphate is not required, so it got accumulated in the form of the granules, and it leads to these granules. That is metachromatic granules. This is how they are formed. Basically, these are the stored phosphate which are present in the form of the granules. Give me a thumbs up. The phosphate is required for nucleic acid synthesis. So whenever nucleic acid synthesis is prevented by starvation, during starvation, no nucleic acid synthesis, no cell division. So the phosphate is accumulated in the form of the granules, and it is the why granules are present. So basically, the intracellular phosphate reserve occurs when nucleic acid synthesis do not take place and granules are seen. So tell me the name of the stains. Tell me the name of the stains by which these granules can be stained. There are 4 stains. इस पे MCQ आता है. The mnemonic is PLAN, P-L-A-N. P stands for Ponder stain. L for Loeffler stain, Loeffler. A for Albert. Albert means the bacteria is green and granules are purple. And N is Neisser stain. So there are 4 stains with the help of which we can stain these granules. We can stain these granules, right? That is the thing. That is the thing. You can see the diagram. You can see the green colored bacteria and the granules at the pole, right? And these are the bacteria in which these granules are present. So there are 5 bacteria जिसमें ये granules हैं. There is a mnemonic. The mnemonic is MSC Garden Agra. Agra में एक garden है जिसका नाम है MSC. I don't know real है कि नहीं, but MSC Garden Agra. M stands for Mycobacteria. S stands for Spirillum volutans, the first bacteria in which it is discovered. That's why known as volutin granules. C stands for Cornybacterium diphtheria, which is our chapter right now. Garden stands for Gardnerella, Gardnerella vaginalis, and Agra stands for Agrobacterium tumefaciens. These are the 5 bacteria in which they are present. Let me summarize the morphology. Let me summarize the morphology. The morphology, I would like to say that I'm teaching you Cornybacterium diphtheria, by the way, right? Cornybacterium diphtheria. These are gram-positive bacilli, right? The bacteria is occurring in the form of the angulated fashion, either in the shape of the L or V, because incomplete separation of the daughter cells. And at the end, they have a blue-purple color granules, which are known as metachromatic granules. Now you get a 2 mark or 3 mark question on metachromatic granules. Metachromatic granules के बारे में सब कुछ बताओ. What you will write if you are getting question on metachromatic granules? What you will write? First, tell me the reason why they are formed, the reason why they are, tell me what they contain, what is their composition, what is their composition, then tell me the reason why they are formed, tell me the name of the stains by which I can stain them, tell me the names of the bacteria in which it is present, और उससे भी पहले tell me its synonym, इसके कितने synonym हैं, how many names you know, how many other names of, so tell me 5 things about it, you will get full marks, 2 mark or 3 mark or 5 mark. Metachromatic granules पे short note चल रहा है, right? So metachromatic granules पे let me write the synonyms. There are 5 names. Metachromatic granules number 1, polar granules number 2, Bates-Ans granules number 3, and volutin granules number 4. The one and the same thing. These are the synonyms. Composition, it composed of phosphate. It contains stored phosphate. Reason, during starvation, the bacteria is not dividing. Since the bacteria is not dividing, no nucleic acid formed. No nucleic acid formed, phosphate is not required. It is not required, so it got accumulated in the form of the granules. So make a flow chart. So that is the reason why they are formed. Coming on the stains. Tell me the name of the stains with which they can be stained. The mnemonic is PLAN. There are 4 stains PLAN. Ponder, Loeffler, Albert, and Neisser. Give me a thumbs up. Tell me the name of the bacteria in which metachromatic granules are present. There are 5 bacteria. The mnemonic is MSC Garden in Agra. In Agra. Mycobacterium, Spirillum volutans, Cornybacterium diphtheria, Gardnerella vaginalis, and Agrobacterium tumefaciens. Give me a thumbs up. So this is a short note on metachromatic granules. Write its synonym, write its composition, write the reason why it is formed, write the name of the stains with which we can stain them, and name the bacteria in which it is present. I want thumbs up from everyone. Jitne audience utni thumbs up. That is a short note on metachromatic granules that you usually get in university exam and on which maximum MCQs also come. In your exam, you may have noticed many MCQs from this portion. So metachromatic granules is done, right? Coming to Cornybacterium diphtheria, right? They are non-capsulated, non-spore forming, non-motile. Na to capsule hai, na to spore hai, na to motility hai. So everything is negative, right? Now you know the mnemonics for all 3. See the mnemonic for capsule. This is the mnemonic for capsule. Keep MCV में C is not Corynebacterium diphtheria. C is Clostridium. It is not, so it is not coming in this mnemonic. So Corynebacterium diphtheria is non-capsulated. It is not coming in this mnemonic. This mnemonic you already know. I have discussed this mnemonic in episode 1 and 2. I'm not reading it again. So apply this mnemonic for all bacteria. If the bacteria is coming in this mnemonic, it is capsulated. If it is not coming in this mnemonic, it is not capsulated. Only 9 bacteria in the world are capsulated. The names are in front of you. So the mnemonic is PACAP, MCV. These are the 9 bacteria which are capsulated. Corynebacterium diphtheria is not coming in this mnemonic. That's why it is non-capsulated. See the mnemonic for spore forming. Spore forming, only 4 bacteria in the world form spore. The mnemonic is BSc chemistry. BSc chemistry में दो C हैं, but none of them is Corynebacterium diphtheria. One C is Clostridium, other C is Coxiella burnetii. It is not Corynebacterium diphtheria, which is my chapter right now. So Corynebacterium diphtheria is non-spore forming. It is not coming in this mnemonic also. So not capsulated, non-spore forming. See the mnemonic for motility. Motility के लिए दो mnemonic हैं. Cute baby sleep, which are motile by Peritracheus flagella, and very protective solution HCl, which are motile by polar flagella. So neither Corynebacterium is coming in this mnemonic, and neither Corynebacterium is coming in this mnemonic. यहाँ पे भी C है, लेकिन C Clostridium है. यहाँ पे भी C है, लेकिन C Campylobacter है. Corynebacterium diphtheria nahi hai, so it is non-motile. So the summary is in front of you. I am teaching you currently the bacteria Corynebacterium diphtheria. So let me summarize till now what I have taught you till now. In the introduction, I taught you the name of the 4 scientists. The name of the scientist who has discovered the disease diphtheria. The name of the scientist who has discovered the bacteria Corynebacterium diphtheria. The name of the scientist who has discovered the toxin of the bacteria. And the name of the scientist who has discovered the antitoxin. All 4 scientists are important. MCQ can come on any of them, right? Most important among them is the name of the scientist who has discovered the bacteria. The name of the scientist is Claps Loeffler. These are the 2 scientists. That's why it is known as Claps Loeffler bacilli, right? Classification, there is no classification. I have not taught you till now. In the morphology, I taught you these are gram-positive bacilli. They occur in angulated fashion. That's why known as Chinese letter arrangement or cuneiform arrangement. At the ends, metachromatic granules are present. So I have given you a summary of the metachromatic granules. You know the name of the stains by which they can be stained. You know the name of the bacteria. You know everything about it, right? These are non-capsulated, non-spore forming, non-motile. This is the summary of morphology in front of you. I want thumbs up from everyone, then only I will proceed ahead. I want thumbs up from everyone. This is the summary of the morphology. Let me come on the culture now. Let me come on the culture now. So Bhatt, Dr. Mahesh, others, have you got it? Give me a thumbs up. Kindly give me a thumbs up in the chat box, those who are watching me live. Can I move ahead? Yes. So let me start with the culture. The culture. So various culture. So I am going to teach you 4 or 5 culture characteristics. The first culture is a liquid medium. The name of the liquid is His serum water. It is a liquid medium. It is a liquid medium. When you grow Corynebacterium diphtheria in His serum water, what you will find? You will find turbidity and pellicle formation at the bottom. Turbidity and pellicle formation at the bottom. You can see the colonies are growing. So the first medium is His serum water. It is a liquid medium. Give me a thumbs up. Learn the name of the His serum water. Most important medium is Loeffler serum slope. Can you see this is Loeffler? It is solid medium. This one is Loeffler serum slope. On Loeffler serum slope, you can see the cream color colonies are formed. So the colonies are creamy white and glistering. So creamy glistering colonies are formed. You can appreciate the cream glistering color. This is Loeffler serum. So the first medium is His serum water. It is liquid. The second is Loeffler serum. The second is Loeffler. The third is the selective media. Selective media are 2. You have to learn the name. First is telluride. Telluride blood agar. On the blood agar, you add telluride. So it becomes telluride blood agar. That is selective. Telluride is a chemical. It is the metal which will kill all other bacteria except Corynebacterium diphtheria. So only Corynebacterium diphtheria will survive on the blood agar containing telluride. All other will die. That's why it is a selective media for Corynebacterium diphtheria. The second is Tinnsdale blood agar. I will tell you composition of that also. So on telluride blood agar contains 0.04% telluride. That inhibits other bacteria except Corynebacterium diphtheria. It contains potassium telluride that kills all other bacteria. Give me a thumbs up. So the color of the colony is black. Can you see it is a blood agar? On the blood agar, please appreciate the color of the colonies of Corynebacterium diphtheria. Corynebacterium diphtheria form black color colonies. Black color colonies on telluride blood agar. Can you appreciate the black color? Give me a thumbs up if you can appreciate the beautiful black color. Give me a thumbs up. These are the black colonies. You can see the black colonies here. So they grow very slowly and black color colonies are formed. The reason for black color, telluride got converted to tellurium. It is an MCQ. Telluride got converted to tellurium. This is the reason for the black color. That's why tellurium is black color. That's why black color colonies are formed. Give me a thumbs up. Everyone give me a thumbs up. Now, based on the telluride blood agar, Corynebacterium diphtheria is divided into 3 different types. Corynebacterium diphtheria का classification मैं अब बता रही हूँ. Now I'm teaching. I have told you na classification. I will tell you later on. So here I am telling you the classification. So on telluride blood agar, we divide into 3 categories: Corynebacterium gravis, Corynebacterium intermedius, and Corynebacterium mitis. Based on the severity of the disease they cause. Gravis cause most severe disease, and this cause moderate disease, and this cause very mild disease. So based on their colony properties, how their colonies are looking on telluride blood agar, uske basis pe I am dividing into 3. Tell me the name of the 3 colonies. Gravis causes serious disease, mitis causes mild disease, and intermediate causes intermediate severity disease. So these are the 3. So gravis form daisy head colony, intermediate form frog egg colony, and mitis form poached egg colony. See the 3 diagrams. See the 3 diagrams. I will explain you, don't worry. So see the gravis here. Gravis. So can you see the diagram of the gravis? It is looking like daisy head. Can you appreciate the colonies are looking like daisy? The colonies are looking like daisy head. Daisy is a flower. You have seen daisy. If you have not seen, Google it out. Daisy head. The head of a daisy. The head of a daisy flower. So the colonies are looking like daisy head in gravis, right? In intermediates, it is known as frog egg. Have you seen a frog egg? The colonies are looking like a frog egg, very small egg. Frog egg appearance colony, right? And in mitis, the third one, the mitis, the colonies look like poached egg. The colonies are looking like poached egg. Please appreciate the colonies are the poached egg. So please learn these 3 things. It is important for university exam also and MCQ also. So which is this medium? The medium is telluride blood agar. Since it is a blood agar, the background is red. Appreciate the red color. This red blood agar contains telluride in that. So it is a selective media for Corynebacterium diphtheria. Only Corynebacterium diphtheria is growing. No other bacteria is growing. It is telluride blood agar. Give me a thumbs up. Give me a thumbs up. So see the appearance of the colony. If the colony are looking like daisy head, it is gravis. If the colonies are looking like frog egg, it is intermedius. And if the colonies are looking like poached egg, it is mitis. Give me a thumbs up. So the summary is that gravis cause very severe disease, intermediate cause intermediate disease, and mitis cause mild disease. So based on the severity of the disease, we are dividing Corynebacterium diphtheria into 3 classes. That is gravis causing severe disease, intermedius causing intermediate disease, and mitis causing mild disease based on the colony appearance. Here colony is daisy head. Daisy is a flower, so daisy head. Here intermedius, it is frog egg. And here it is poached egg. Give me a thumbs up. So I've shown you the diagrams also. I'm expecting that you got it. If you got it, kindly confirm by giving a thumbs up. So daisy head, frog egg, and poached egg. Gravis, intermedius, and mitis. So learn like that. This is a beautiful diagram showing the 3. So again, see the beautiful diagram showing the 3. It is daisy head, it is frog egg, it is poached egg. Please appreciate it. Daisy head, frog egg, poached egg. It is an MCQ also and an exam also. You have to write the 3 things. Give me a thumbs up. Give me a thumbs up. That was the first selective media, telluride blood agar. The second selective media is Tinsdale agar. So Tinsdale agar, we add cystine. Along with telluride. Blood agar में telluride तो है ही. Additionally, हम सिर्फ एक चीज और डाल रहे हैं, cystine. So now the telluride agar converted into Tinsdale agar. It is more selective. Here also black color or brown color colonies are formed. You can see a black color colony. Appreciate the black color colony which is surrounded by a brown halo. Black color colony surrounded by a brown halo. Black colony surrounded by a brown halo. That's it. That's it. Let me summarize the culture. I am going to teach you 3 culture. 1, 2, 3. Tell me the liquid culture. The name of the liquid culture is his serum water. In his serum water, turbidity is there and pellicle formation is there. The second media, the solid media is Loeffler's serum slope. On Loeffler's serum slope, cream color glistering colonies are formed. And third one is the selective. Selective mein I am going to teach you 2. The first one is the telluride media, and second one is the Tinsdale. Tinsdale. Learn the composition of the two. Telluride contain blood agar along with potassium telluride. Potassium telluride is there. In Tinsdale, blood agar plus potassium telluride plus addition of cysteine. Give me a thumbs up. Everyone give me a thumbs up. Based on the telluride agar, there are three types of corynebacterium: Gravis, Intermedius, and Mitis. Based on the colony appearance. In Gravis, the colony appearance is daisy head. In Intermedius, the colony appearance is frog egg. And in Mitis, the colony appearance is poached egg. Give me a thumbs up. This is the summary of the culture. One is liquid, one is solid, and two are selective. So total 4 media should be at the tip of the tongue. Everyone give me a thumbs up. Everyone. You also, Madmix, you want a lecture on genetic disorder? Definitely. So in the next month, that is in June, definitely in my deliverables, I will arrange some of the lecture on genetic disorders. Don't worry. So on YouTube only, I will arrange that. Infectious diseases too. So infectious diseases only I am teaching right now, Madmix. Which other infectious disease you want? So kindly enumerate that. I will try to arrange that lecture if maximum students are interested. Okay. So we are done with the culture. Now, I am coming on the biochemical reaction. Culture is clear to you? Till now, we have done the introduction, the name of the 4 scientists who has discovered the bacteria, who has discovered the disease, who has discovered the toxin, and who has discovered the antitoxin. So, four scientists are clear, right? After that, classification मैंने यहाँ पे बताया. Gravis, Intermedius, Mitis. We know the classification. Morphology, you know the morphology. These are gram-positive bacilli. They are in the form of the angulated fashion, that is V or L. So Chinese letter arrangement, and at the poles they have a metachromatic granules. You know what are metachromatic granules. These are non-capsulated, non-spore forming, and non-motile. We know that. After that, culture is in front of you. Now I'm coming on biochemical reactions. Shall I start biochemical reaction? 5 biochemical reactions are important. I will discuss all of them one by one. So 4 culture media and 5 biochemical reaction. I'm teaching you the 5 biochemical reactions like this. See, everyone here, see, I'm teaching you the 5 biochemical reactions. So the first biochemical reaction is fermentation of sugars. They ferment sugar. So second is H2S positive. Third, they reduce nitrate to nitrite. They are urease negative, which differentiate them from other bacteria. It is a negative test. And they are PYZ positive. PYZ is pyrazinamidase positive. So first is fermentation of sugar. They ferment glucose and maltose with the production of acid but no gas. They do not ferment lactose, mannitol, and sucrose, other sugar. They ferment only glucose and maltose, the 2 sugar, with acid, without gas. They are H2S positive. They reduce nitrate to nitrite. They are urease negative, and they are PYZ positive. You know, you have to enumerate the biochemical reaction like this. Don't go in details. Just describe them in one word, in one line. What are the 5 type of biochemical reactions? They are first is sugar fermentation. In sugar, they are fermenting glucose and maltose, not other sugars, with acid, without gas. Right, they are H2S positive. They reduce nitrate to nitrite, right, and they are PYZ positive. Urease negative, you can skip, right. These are the 4. So 4 culture, 4 biochemical reactions. Everyone give me a thumbs up till now. Let me come on the antigenic structure. In the antigenic structure, it is having 2 type of antigen, O and K. O antigen and K antigen. Based on the O and K antigen, which is present in the cell membrane, cell wall, they are of various types. So Gravis is of 13 types, Intermedius is of 4 types, and Mitis is of 40 types. So based on that, resistance is not really important. Antigenic structure, you can resistance, you can skip, directly come on the virulence factor. The most important virulence factor is toxin. It produces a toxin, the most lethal toxin, diphtheria toxin. Let me describe the diphtheria toxin. इस पे अलग से short note आता है. Write a short note on diphtheria toxin. Virulence factor में toxin. The name of the toxin is diphtheria toxin. So I am giving you a short note on diphtheria toxin. Write everything about the diphtheria toxin you can. So I am teaching you the toxin, the virulence factor of the Corynebacterium diphtheria. So toxin is an exotoxin. You know, there are 2 type of toxins. There are some bacteria, they are having cell wall. This is another bacteria, this is the cell wall. Sometimes toxin is present as a component of the cell wall. This is endotoxin. And sometimes toxin is present inside the bacteria, but it's secrete outside. This is exotoxin. This is exotoxin. This is endo. This is exo. So diphtheria toxin is an exotoxin. It is not an endotoxin. It is an exotoxin. Give me a thumbs up. It is an exotoxin, right? And this toxin is necessary for the virulence. If any bacteria, Corynebacterium diphtheria, causes diphtheria is the disease caused by Corynebacterium diphtheria, then it must produce toxin. Without toxin, it cannot produce disease. So for toxin production, for disease production, for causing disease, for causing pathogenicity, toxin is must. So avirulent strains, that is, they do not cause disease. They do not cause, they do not contain toxin. They cause only carrier. They are present only in carrier, not in cases. You got my point. So toxin production ke liye there is a gene which is required. The name of the gene is tox gene. The name of the gene is tox gene, and there is a phage. Beta phage is the virus which transmit this gene from one bacteria to another. You got my point? Listen, see, this is Corynebacterium diphtheria. This is the nucleus of the Corynebacterium diphtheria, and this is the bacteria Corynebacterium diphtheria. In the nucleus, this is the chromosome. On the chromosome, a gene is present which is known as tox gene. With the help of tox gene transcription and translation, this toxin is formed in the cytoplasm, and it is exotoxin. It is responsible for causing the disease diphtheria. It is responsible for virulence. It is responsible for causing this disease diphtheria. Now imagine another bacteria. It is also Corynebacterium diphtheria, but the chromosome do not contain tox gene. Since tox gene is absent, it do not contain toxin. That's why this one is avirulent. This one is virulent. This one is avirulent. It do not causes diphtheria. Now there is a virus. The name of the virus is beta phage. Beta phage. The name of the virus is beta phage. This virus is transmitting the tox gene from this bacteria to this bacteria. It is like a carrier. It is transmitting the tox gene from this bacteria to this bacteria. So avirulent strain can be converted to virulent strain. It will receive the tox gene and it will start producing toxin, the exotoxin. Now avirulent strain can be converted to virulent strain with the help of the beta phage, and this mechanism is known as lysogenic conversion. So can you write a short note on lysogenic conversion? What is, you can draw this diagram. So basically this is happening in lysogenic conversion. In lysogenic conversion or phage conversion, there is a virus. The name of the virus is beta phage that is causing transmission of the tox gene. So non-toxigenic strain can be converted to toxigenic strain. Avirulent bacteria can be converted to virulent bacteria with the help of beta phage, and this is known as lysogenic conversion. Give me a thumbs up. The name of the gene is tox gene. You can see the same diagram which I have shown you. You can see here beta phage is doing the transmission, and this gene is tox gene. Can you see? This is diphtheria toxin gene, the tox gene. It is transmitted from the bacteria, one bacteria to another with the help of a phage, that is beta phage. So this is a virus. It is coming here, taking the tox gene from here and transmitting to another bacteria. So non-toxigenic strain can be converted to toxigenic strain, and this is known as lysogenic conversion. I want your appreciation that you got my point. You got my point? Yes or no? Yes or no? So this is how we convert a non-toxigenic strain to a toxigenic strain. You see, this bacteria do not contain tox gene. When the phage is coming, it is giving the tox gene. So tox gene is inserted. Now it contains tox gene. So initially it was avirulent, non-toxigenic. But now on receiving the gene, it converted into toxigenic strain and it become virulent. So this conversion is known as lysogenic conversion. This is how I can explain you the lysogenic conversion. Now coming on the properties of the toxin. The toxin ka weight is 62,000. This toxin is having two subunits, two fragments, fragment A and fragment B. Individual weight don't learn. Total weight is sufficient. Fragment A and fragment B, both fragments are required for toxigenicity. The toxin is having two fragments. Fragment A has enzymatic activity and fragment B is responsible for binding. B for binding with the cell, right? B for binding with the cell. It is heat labile. Once you heat the toxin, it becomes inactivated. It is heat labile, but it is extremely potent. See the potency. 0.0001 milligram can kill a guinea pig. Can kill a guinea pig. It can kill. It is lethal for that. Humans के लिए थोड़ा सा ज़्यादा dose लगेगा. A little bit more dose can be lethal for humans. So it is very potent and lethal toxin. Give me a thumbs up. Give me a thumbs up. Now this is toxin. Toxin. It is active. It can produce the disease. Once the toxin become inactive, it is known as toxoid. So toxin is active. It can produce the disease. Once it become inactive, it is known as toxoid. Toxoid is required for vaccine production. We do inactivation of the toxin and fill it in a vaccine. So vaccine for diphtheria contains toxoid. That is inactive toxin, right? So how we do the conversion of toxin to toxoid? Either by heating or by treating it with formalin or with acidic pH. So toxin can be converted to toxoid. That is, active toxin will become inactive toxin. Inactive toxin is toxoid by three mechanisms. Either by heating or by treating with formalin or giving acidic pH. Acidic pH. By these three mechanisms, we can convert toxin into toxoid. And this toxoid can be Toxoid is a toxin which loses toxicity. It is inactive, but it is having antigenicity. So it is capable of producing still antitoxin. It is used for vaccine purpose, right? And iron is required for toxin production. Iron is must. Iron is must, right? That is the all important points. This toxin loves 3 organs in human body. After going in the blood, the toxin go to the 3 organs in human body. Number 1, heart, myocardium. Number 2, adrenal gland. Above the kidney, there is a gland, adrenal gland. And number 3, nerves. From the brain, the nerves are coming, sympathetic, parasympathetic nerves. So number 3, nerves. So heart, adrenal, and nerves. These are the 3 organs. The toxin will go and damage these 3 organs. So in diphtheria, these 3 organs are damaged. Diphtheria disease. Got my point? Mode of action. This toxin inhibit protein synthesis inside the cell. Once it go inside the cell, the fragment A, fragment B will bind with the cell. Suppose this is a cell. This is a human cell. This is a host cell. Host means human. Either it is a heart cell, that is myocardium, or it is the adrenal cell, or it is a nerve cell. Toxin love these 3 cells. And this is toxin. In the toxin, this is fragment B and fragment A. With the help of fragment B, it is binding with the human cell. After that, the toxin enters inside the human cell. In the human cell, now fragment A will show its activity. It is showing enzymatic activity, and it split NAD to ADPR. It split NAD to form nicotinamide and adenosine diphosphoribase ADPR. This ADPR will inactivate elongation factor 2. Elongation factor 2 is required for protein synthesis and transcription and translation on the ribosome. So protein synthesis do not take place. So imagine I'm telling you again. Listen, this is a human cell, host cell. Usually it can be any cell, but usually either it is myocardium or it is a nerve cell, or the third one is the adrenal, because toxin love these 3 cells in the human body. So after coming in blood, toxin have 2 fragments, fragment B and fragment A. B is for binding. The fragment B is binding with the cell, and after that the toxin enters inside. Now there is no role of B. A convert NAD into ADPR. ADPR. This ADPR inhibit the elongation factor 2. So there is no elongation factor 2 inside these cells. So inside these cells, there is no protein synthesis. Since there is no protein synthesis, these cells will die. These cells will die, and there is disease in the 3 cells. There is myocardiopathy, there is neuritis, adrenal maybe disease hoga. So various diseases will occur. The organs will be damaged. So this is the mechanism of action of the toxin. Can you see the same diagram is shown? If you can appreciate, this is fragment A and B. B is required for binding on the receptors, right? After that, toxin is entering inside. After entering inside, it is converting NAD to ADPR. ADPR is inhibiting elongation factor 2. So no protein synthesis, the cell will die. Give me a thumbs up. Everyone give me a thumbs up. 4 toxins act by similar mechanism. First one is the diphtheria that I have taught you. After that, Shiga toxin in Shigella, Shiga-like toxin in E. coli and Pseudomonas. So diphtheria, Pseudomonas, Shiga and Shiga-like. These are the 4 toxins. All of them act with the same mechanism. They inhibit protein synthesis in the cell and degrade the cell. So any question coming in your exam, either the mechanism of diphtheria toxin, the mechanism of Pseudomonas toxin, the mechanism of Shigella toxin, or the mechanism of Shiga-like toxin in E. coli. Draw this flowchart, draw this diagram. So that is the thing. Give me a thumbs up. The mechanism is same for all of them. Once for all, मैंने एक ही बार में सबका बता दिया. Give me a thumbs up. Tell me virulence factor. The virulence factor, only one virulence factor is there, that is toxin. Tell me everything about toxin. Toxin पे short note बताओ. Tell me short note on diphtheria toxin. Who will tell me the short note on diphtheria toxin? Who will tell me? Tell me the molecular weight. Tell me it is having 2 fragments, fragment A and B. Both are required. B for binding and A is having enzymatic activity. It is very lethal. So even 0.0001 milligram is sufficient to kill a guinea pig. It is very lethal. Iron is required for its synthesis, right? Iron is required for its synthesis. The mechanism of action is protein synthesis inhibition. It does protein synthesis inhibition. You know the complete flowchart. It acts on 3 organs in human body. It acts on myocardium, the nerves, and adrenal, right? So you have to write everything about the toxin, whatever you know. Give me a thumbs up. Everyone give me a thumbs up. So that is the diphtheria toxin. You got it? Say yes or no. So that is a short note on the diphtheria toxin. It is the most potent lethal toxin. So diphtheria toxin is done. Toxin can be converted to toxoid by 3 mechanisms. You already know the 3 mechanisms: heating, formalin, acidic pH. That also you can write. Coming on the pathogenesis. Name the disease caused by Corynebacterium diphtheria. Corynebacterium diphtheria causes only one disease, that is I'm sorry. It causes only one disease, that is diphtheria. The name of the disease is diphtheria. As the name indicates, Corynebacterium diphtheriae causes one disease, diphtheria. But diphtheria having various things, local effects, systemic effects, and complication. We will be discussing them one by one, right? Diphtheria, bacteria do not go inside human body. Only toxin of diphtheria will go inside the body. That's why diphtheria is not septicemia or bacteremia. Diphtheria is toxemia. Toxemia means toxin in blood. The meaning of the toxemia is toxin in blood. There is no bacteria in blood. Toxin in the blood will cause diphtheria. So it is a line you have to learn. Diphtheria is not bacteremia. Diphtheria is not septicemia. Diphtheria is toxemia. The toxin of the diphtheria is causing diphtheria. That name, the disease diphtheria. Diphtheria usually occurs in children, 2 to 10 years. So humans are the only reservoir. It is confined to humans only, and droplet infections, it is spreading from one person to another. There are cases, there are carrier. Cases are the persons जिसमें symptoms हैं. Carrier are the persons who do not have symptoms, but they are transmitting the disease from one person to another. The ratio of cases and carrier, 95 are the carrier and only 5% are the cases. You know, the same problem we faced in COVID. In COVID also we have abundant of carrier. So it is difficult for a disease to treat when carriers are more than cases. Because carriers are invisible now. They do not have any symptoms, but they are transmitting the disease in the society, in the community. So carriers यहाँ पे 95 हैं, only 5% carriers. For every 5 cases, there are 95 carriers. That's why diphtheria is a disease which cannot be eliminated. इसको eliminate हम कर ही नहीं सकते because most of them are not cases. यदि case हो तो we can treat it, but carrier we cannot treat a carrier because it is invisible. It is invisible, and carriers are of two types: nasal and throat. Like COVID only, the bacteria can be present in the nose, the bacteria can be present in the throat. So while coughing and sneezing, the bacteria comes in the air. So imagine this is a person. Don't laugh on my diagram, please. I'm poor at drawing. So the bacteria can be present in the nose, it can be present in the tonsils and the throat. So while coughing and sneezing, it is coming in the air in the form of the droplet, and it is spreading. It is spreading from one person to another. This is known as droplet infection. This infection is known as droplet infection. Give me a thumbs up like COVID only. So nasal are more dangerous than throat. Nasal carriers, the two type of carriers, nasal carriers and throat carriers. Give me a thumbs up. Give me a thumbs up. Sometimes the bacteria present in the tonsil. If it is present in the tonsil, it is known as faucial. Sometimes the bacteria present in the larynx, it is known as laryngeal. Sometimes it is present in the nose, it is known as nasal. Sometimes the bacteria present in the conjunctiva, in the ear is otitic. Sometimes in the vulva and vagina of the females, and sometimes in the skin. So bacteria can be present anywhere. So most common is the faucial, they are present in the tonsil. So commonly tonsil hai, lekin dangerous nasal hai. So coming on the three type of effects one by one. Let's start with local effects. What is the summary? The bacteria will go, multiply, start producing toxin, and bacteria and toxin together causes necrosis of that organ and produces a pseudomembrane over it, which is made up of fibrinous exudate, leukocyte, and erythrocyte. I will tell you. And this is the local effect. Okay. Let me draw the two tonsils of a person. Okay. Let me draw a person for you. Let me draw a human being for you. Let me draw the two tonsils of this person. Right. Through droplet infection, the bacteria entered inside. This is Corynebacterium diphtheriae. Corynebacterium diphtheriae entered inside through droplet infection. After that, it is going on the tonsil. In the tonsil, they will multiply. The bacteria, they are not going in blood. Mind my words. After multiplying, they produce toxin. Now, bacteria and toxin together causes necrosis of the organ. They causes necrosis of the tonsil, and necrotic debris are produced. The necrotic debris along with WBC, along with RBC, along with necrotic debris, they will form a membrane-like structure, but it is not a real membrane. It is a pseudomembrane. So a pseudomembrane, this tonsil is covered with a pseudomembrane. This pseudomembrane is made up of necrotic debris, RBC and WBC. And it is not a real membrane, that's why known as pseudomembrane. Why it is known as pseudomembrane? Once you try to remove it, if it is a real membrane, you can remove it, separate it from the underlying tonsil. But once you try to remove it, you cannot separate it. The tonsil will start bleeding. You cannot separate it. That's why it is a pseudomembrane. A membrane, a white color membrane is visible. It is known as pseudomembrane. Give me a thumbs up. Can you appreciate a pseudo white color membrane here over the tonsil? This is pseudomembrane. This is local effect. Till now, bacteria or toxin do not enter inside the blood. So bacteria is going inside the tonsil, they are multiplying, they are producing toxin, Bacteria and toxin together causes the necrosis and it produces a pseudomembrane. This is the local effect. Patient has severe throat pain. Severe throat pain, it is the local effect. Give me a thumbs up. If you try to dislodge the membrane, the patient bleed. The patient bleed. Can you see? This is the pseudomembrane. If you try to dislodge it, the patient will bleed. That's why it is known as pseudo membrane. It is not a real membrane. Sometimes the lymph nodes get enlarged here. The lymph nodes, it is known as bull's neck. The neck is like a bull's neck because of the enlargement of the lymph nodes in the neck. This is the local effect. In the local effect, two things are there. The lymph node get enlarged, so known as bull's neck, and the pseudomembrane is there. These two things together known as local effect. Locally they are producing. They are producing this effect in the tonsil if they are in tonsil. The same effect can be produced in the nose if they are present in the nose. Nose में pseudomembrane बनेगा. Whatever is the organ of the entry, वहाँ पे pseudomembrane बनेगा. That is local effect. Local effect. So you can see the bull's neck. You can see the pseudomembrane. Together they are known as local effect. Local effect में दो चीजें हैं, pseudomembrane and bull's neck. Give me a thumbs up. Give me a thumbs up. So that is local effect. Local effect is due to toxin and diphtheria bacteria together. They both are causing the necrosis, right? So local effect are there. Coming on systemic effect. Now, toxin will enter inside the blood. Bacteria will not enter inside the blood. What I'm telling you, let me draw the same human again. These are the tonsils. So this is the bacteria present here. Bacteria produce toxin. So this is the toxin produced by the bacteria. Bacteria and toxin together causes necrosis and produces a pseudomembrane over the tonsil. That is local effect. That is done. Now, this is the blood vessel of this human. In the blood vessel, only toxin is entering. Bacteria is not entering. Mind my words. So it is toxemia. It is not bacteremia. It is toxemia. Please mind it. Please mind my words. So only toxin of the diphtheria, Corynebacterium diphtheria, is entering inside. The bacteria is not entering inside. It is known as toxemia. Give me a thumbs up. So systemic effects will be due to toxemia, right? So the diphtheria toxin going inside in the blood. In the blood, it is producing toxemia. After going in the blood, this toxin is going to the three organs: heart, adrenal, and nerves, and damaging the three organs. What are the three organs? Heart, adrenal, and nerve. And they causes damage to these three organs. There is no bacteremia. There is toxemia. Give me a thumbs up. So in systemic effects, the three organs are damaged like this with the help of the toxin. So local effects are produced by bacteria plus toxin both. That is necrosis. But systemic effects are produced only by toxin. You got my point? Nisha, Raj, Che, you got my point. Others, you got my point. So it is toxemia. Coming on the complications. Complications can be mechanical due to pseudomembrane. Pseudomembrane की वजह से mechanical. So if imagine the point of entry is larynx. I have told you point of entry can be anything. It can be nose, it can be tonsil, it can be throat, it can be larynx, it can be vulva vagina, it can be skin. If it is larynx, so at the point of entry, pseudomembrane is formed. So pseudomembrane will be formed at the larynx, so patient can't breathe. Larynx inlet will be closed with the help of pseudomembrane, and patient can't breathe, patient can die. Yes. So mechanical complications can be there because of the pseudomembrane, right? So patient can have asphyxia if the respiratory passage or larynx is blocked. In emergency, you have to do a tracheostomy to save the patient, right? If heart is involved, patient will go in acute circulatory failure. If myocardium, the heart is involved. If nerves are involved, patient will have paralysis. If the nerves are involved, post-diphtheria paralysis. I told you three organs, na? Adrenal, heart, and nerves. Heart is involved, patient will go in circulatory failure. And the nerves are involved, patient will go in paralysis. How many cranial nerves are there? I guess there are 12 cranial nerves. You already know the names. So the most common cranial nerve which is involved is ocular. Ocular cranial nerve most commonly isolated. It causes paralysis of the third cranial nerve, that is ocular, right? And palatine and ciliary branches are involved, but not pupillary paralysis, right? Spontaneous recovery is there for the paralysis, right? So most common complication is myocarditis due to the involvement of the heart. Second most complication is neuritis because of the involvement of the nerves. Everyone give me a thumbs up. Now, can you give me the summary of the three effects? So, Corynebacterium diphtheria causes only one disease, diphtheria. So, diphtheria have local effect, systemic effect, and complication. Tell me the three things. So, as soon as the bacteria enters human body, it can enter via various routes. It enters via droplet infection, and carrier are more than cases. 95:5 is the ratio between carrier and cases, right? Via droplet infection, it will go to the tonsil. It will multiply there. It will produce toxin. Bacteria and toxin together causes necrosis of the organ, and necrotic debris along with WBC and RBC will produce a membrane known as pseudomembrane. So pseudomembrane is the end result of local effect. and that is produced by bacteria as well as toxin. That's it. Now, only toxin enters in the blood, not bacteria. Only toxin, producing toxemia. This toxin will go to the three organs. It will go to the adrenal, it will go to the heart, it will go to the nerves and damages these three organs, causing the systemic effects. Patient have fever and all these things, right? After that, coming on the complications. In the complications, mostly there are mechanical complications due to the pseudomembrane. It can be laryngeal obstruction, it can be throat pe pseudomembrane, nose pe pseudomembrane. So mechanical obstruction. If heart is involved, carditis is there, circulatory failure is there. If nerves are involved, paralysis is there. Post-diphtheria paralysis. Paralysis can be usually of the ocular nerve. You know the ciliary and palatine branches are involved. Pupillary is not involved. Pupillary branch is not involved. So you know the branches of the ocular nerve. Everyone give me a thumbs up. So this is a summary of the diphtheria, the pathogenesis diphtheria. Everyone give me a thumbs up. You got my point? So we are done with the pathogenesis. In the pathogenesis, write down diphtheria. Write down the three things in the diphtheria: the local effect, the systemic effect, the complications. Local effect is due to bacteria, systemic effects is due to toxin, and complication can be due to of anything. Everyone give me a thumbs up. I tried my best, right? Coming on the lab diagnosis. For lab diagnosis, first tell me the specimen. जब भी lab diagnosis शुरू करो, पहले specimen बताओ. You will say, ma'am, depending, specimen will depend. What is the point of entry? Where is the pseudomembrane formed? If the pseudomembrane is formed over the throat, over the nose, larynx, ear, conjunctiva, vagina, skin, we will take the pseudomembrane. So pseudomembrane is swab. We will take swab from the pseudomembrane. Like in COVID, we take two swab, one from the nose, one from the throat. Usually it is nose and throat. So we will take the swab from the pseudomembrane. The pseudomembrane, we will take the swabs. That is the specimen. Now do direct microscopy. You will find Chinese letter arrangement. You already know. Do the culture. In the culture, do the selective media. You do the culture on telluride media, black color colonies, or Tinsdale media. That is black colonies with brown halo. Give me a thumbs up. Do the biochemical reaction. In the biochemical reaction, they are H2S positive. They are PYZ positive. You already know the biochemical reaction. Coming on the virulence test directly. You already know the first three things. Shall I skip that? The first three things you already know. So direct microscopy, you do Gram stain, you do Albert stain, you will find the metachromatic granules with Chinese letter arrangement. You already know, I have described that, right? So that is the organism. Culture pe you already know Loeffler serum pe cream color colonies are formed. Telluride pe black color colonies are formed with green halo. And you already know the Tinsdale medium, black colonies with brown halo. Biochemical reaction में fermentation of sugar, you already know it ferment glucose and maltose but not other sugar. H2S is positive, nitrate to nitrate reduction, urease negative, and pyrazinamide positive. Coming on virulence test. For doing virulence test, we will do two tests: in vivo, in vitro. What do you mean by in vivo, in vitro? Now first let me tell you in vivo. In vivo is easy, but in vitro is the most important one for your exams. In vivo you will do on guinea pig. In vivo, we will do it on guinea pig, subcutaneous and intracutaneous, we will give on guinea pig. But in vitro, the most important is Alex gel precipitation test. In vitro. In vivo, yani means inside living organism. In vitro means outside living organism. That is in vitro. Yes or no? So let me teach you first in vivo. Take two guinea pigs. Inside one guinea pig, directly give diphtheria toxin. In the second guinea pig, first give antitoxin protection. That is vaccine. Vaccine is antitoxin. First give antitoxin protection, the pre-made antitoxin, not exactly vaccine, the pre-made antitoxin, right? The passive immunity, followed by give diphtheria toxin. You will notice the guinea pig in which you are directly giving diphtheria toxin, the guinea pig will have disease. It will have diphtheria, the organs, the adrenal, the nerves and the heart will damage, and the guinea pig will die within few days, right? That is having diphtheria. But the guinea pig inside which you are initially giving protection with antitoxin will not die. It will remain alive. Here you are giving both of them the injections via subcutaneous route. Give me a thumbs up. Very easy experiment in vivo. Everyone. So take two guinea pigs. Inside each of them give 0.5 ml of injection, subcutaneous injection of the toxin. But in one of them, give 500 units of antitoxin previously. You will find what is the interpretation. The dead one autopsy जब आप करोगे, the dead one का autopsy पे, you will find the adrenal gland is hemorrhagic, the myocardial damage is there. You know, all the organs are damaged like that, right? And the other one will remain alive. So that is subcutaneous. Same experiment can be done with intracutaneous route. Same can be done with intracutaneous route also. यहाँ पे एक को directly toxin देंगे, एक को एंटीटॉक्सिन फॉलोड बाय टॉक्सिन. So जिसको डायरेक्टली टॉक्सिन दिया that will die, and जिसको एंटीटॉक्सिन फॉलोड बाय टॉक्सिन दिया that will not die, that will remain alive. Here actually intracutaneous में die नहीं होगा, इसको सिर्फ disease होगा. So here organism is sacrificed. That's why in vivo are not used generally. They are only for theory purpose. But you should know in vivo test होते हैं. Coming on vitro test. In vitro test में the most important is Alex gel precipitation test. Here I will prove the virulence. Here we will prove the virulence. In vitro test, Alex gel precipitation test, it was discovered by Alex. That's why Alex gel precipitation test. Basically, what you are doing, you are taking a agar plate. Okay. Okay. In the agar plate, okay. Just a second. Okay. Okay. Got it. So you take a agar plate. You take a blood agar plate. Take a rectangular strip. This is a rectangular filter paper. It is soaked in antitoxin. It contains antitoxin. It already soaked in antitoxin. It is dipped in antitoxin and placed over the agar plate. This is antitoxin. Everyone give me a thumbs up. It's very easy. Now take your test strain. Whatever is your test strain. I want to test this strain, whether this strain produce toxin, yes or no. So I will give a perpendicular streak of my test strain. This is my test strain of the bacteria. I'm giving a perpendicular strip over the antitoxin, right? Now, I don't know whether it produces toxin or whether it do not produce toxin. I want to know that only, whether it is virulent or avirulent. If it produces toxin, it is virulent. If it do not produce toxin, it is avirulent. I want to know that only. Imagine it produces toxin. So where it will produce toxin, the toxin will come here, here, here. It start producing toxin because it is toxigenic strain. So it will produce toxin around it. It will produce toxin around it, and antitoxin will diffuse. Antitoxin is present in the filter paper here. The antitoxin will also diffuse out here. The antitoxin will also diffuse out. So toxin antitoxin reaction takes place, and here lines are formed. These are the lines. These are the lines of precipitation. So it is an example of precipitation. Give me a thumbs up. So you can see the lines of precipitation at the junction of toxin and antitoxin. If lines of precipitation are formed, means the test strain is producing toxin. If lines of precipitation are not formed, means test strain is not producing toxin. So whether the test strain is toxigenic or non-toxigenic, virulent or avirulent, that can be proven like this. Give me a thumbs up. Everyone give me a thumbs up. So take a test strain here. The test strain, take a filter paper which is soaked in the antitoxin. Now take your test strain. Now before taking the test strain, you should take the positive and the negative control. You know, so you are taking a agar plate. On the agar plate, you are taking the antitoxin. The filter paper soaked in antitoxin. This is antitoxin. Now take first strain. The first strain is a positive strain. Positive strain means you know that it produces. You are sure that it produces toxin. It is a positive strain, it produces toxin, right? Take a negative strain. Take a negative strain also for the control. These are the two controls. You know that it do not produce toxin. You are sure it do not produce toxin. This is a negative strain. And third strain is the test strain. For test strain, I don't know whether it produce toxin or not produce toxin. You got my point? So this definitely produce toxin. So you will find the lines here. And this definitely do not produce toxin. So you will find no lines here. In the test strain, if both of them are working, positive strain is also working, negative is also working. You are finding the lines of precipitation around positive strain, and you are not finding the lines of precipitation around the negative strain. Then whatever is the result of your test strain, you are sure. Now test strain के around line आ भी सकते हैं, नहीं भी आ सकते हैं. If they are coming, means it is toxigenic. If it is not coming, it is non-toxigenic. Imagine you are not taking positive and negative strain for the control. You say कि मैम control की क्या जरूरत है? I am taking directly the test strain. आपने directly test strain ले लिया, right? Imagine you are saying, ma'am, I will not take the control. I will directly test on the test strain. So this is my antitoxin and this is my test strain. I'm taking it directly. This is my test strain. So if it is negative, it is not producing toxin, you will not get lines of precipitation. So you will get confused. Actually it is negative or you have done some mistake during the procedure. That's why it is not showing the result. So taking control is always advisable. Give me a thumbs up. So take a positive control. Positive and negative controls are important. Give me a thumbs up. Can you see this one is positive control here? The first line is positive control. You can see the lines of precipitation around it. You can see the third one is the negative control. So no lines of precipitation around it. And the middle one is the test strain. The test strain is also showing positive, that is lines of precipitation. So it is toxigenic. So lines of precipitation are important. Give me a thumbs up. So this is Alex gel precipitation test. See the two bacteria in the diagram. This is antitoxin ka strip, right? Here, the strain. I have taken first bacteria here, the second bacteria here. The two bacteria are bacteria A and bacteria B. Bacteria A ke around, I am getting the lines of precipitation. Bacteria B ke around, I am not getting the lines of precipitation. So my summary is that bacteria A is toxigenic or virulent, and bacteria B is non-toxigenic or avirulent. I cannot simplify more than this. Everyone give me a thumbs up. Toxigenic strain produce line of precipitation. Non-toxigenic strain do not produce lines of precipitation. This is the result. Write the principal, method, result. For all tests, write the principal, method, result. I'm done with Alex gel precipitation test. Others are not really important. You can skip them. I am done with the lab diagnosis. In the lab diagnosis, most important is the virulence test, in vivo, in vitro. In vivo done on the guinea pig, and in vitro is the Alex gel precipitation test. Coming on the treatment. Okay, just a second. Okay. Schick's test, I'm skipping here the Schick's test. Coming on the treatment directly. I'm skipping the Schick's test here. Coming on the treatment. What is the treatment of the diphtheria? Who will tell me the treatment? So what is the treatment of the cases? What is the treatment for the carrier? The treatment for the cases, if any case is there, give diphtheria antitoxin. The first treatment is the antitoxin. You know, I have told you that this is a human. Inside the human, these are the tonsils of the human. What is the process? And this is the blood vessel. And these are the organs. This is adrenal, this is heart, and these are the nerves. So what is the complete mechanism? Who will tell me? Nisha, would you like to tell? So first the organism, the bacteria, the diphtheria, Corynebacterium diphtheriae enters from the droplet infection. It will go on the tonsil, multiply on the tonsil and produce toxin there. Because of the bacteria and toxin together, here a pseudomembrane is formed. This is the local effect. Because of the necrosis, pseudomembrane is formed. Now, mind my words, toxin enter in the blood. Only toxin, not bacteria. Toxin enter in the blood and damages the three organs. That is the systemic effect. Yes or no? So for treatment, I'm giving you two options. Now you yourself tell. For treatment, you want to give antibiotic or antitoxin. What is main treatment? Antitoxin. Because only toxin is entering in the blood, the main treatment is antitoxin. You also give antibiotic. Antibiotic will act locally on the tonsil only. Yes or no? Yes or no, Nisha? So it will act only on the tonsil. Yes. So antibiotic kill the bacteria locally. Antibiotic is important, but antibiotic will not act in the blood because in the blood there is no bacteria. Bacteria is present. Antibiotic kills bacteria, and antitoxin will neutralize the toxin. Right? So toxin is present both ways. So antitoxin यहाँ भी काम करेगा, यहाँ भी काम करेगा. It will act both. But antibiotic act only locally. So give both, but mainstream of treatment will be antitoxin, not antibiotic. Give me a thumbs up. So first give antitoxin. Start with antitoxin. Learn the doses. Start with antitoxin. Depending on the severity, you decide your dose. Yes, Joy. Yes, Nisha. Absolutely right. Go with antitoxin first. But give antibiotic also. So antibiotic may either give penicillin or erythromycin, right? So that you can also give. So give two things, antitoxin, antibiotic. But main treatment here is antitoxin, not antibiotic. Antibiotic also you can give, but not the primary treatment. For carriers, now coming on the carrier, who is a carrier? Who is a carrier? Carrier is a person. This is the blood vessel and these are the organs. This is the kidney, this is the heart, these are the nerves. The same diagram I'm drawing. In the carrier, through droplet infection, the bacteria is entering and present on the tonsil. And this is asymptomatic. It is not causing pseudomembrane here. It is asymptomatic. The bacteria is present, but bacteria is not producing toxin. Mind my words, I'm talking about carrier. Who is the carrier? In the carrier, the bacteria is present, but bacteria is not producing toxin. Since toxin is not produced, that's why it is asymptomatic. That's why it is asymptomatic. So carrier mein what is the treatment? You tell me only. Common sense. Now, what is the treatment in a carrier? Would you like to give antitoxin here? No role of antitoxin. Here only antibiotic is given. For carrier only antibiotic is given. Yes, Nisha? You got it? Joy? You got it? For carrier give only erythromycin. No antitoxin is required. What is the treatment of cases? What is the treatment of carrier? Who will tell me the summary? Treatment of case is antitoxin followed by antibiotic. Antibiotic is penicillin or erythromycin. Here treatment is only antibiotic. Yes or no? Yes or no? You got it? You got it? That is the treatment. Treatment is very, it based on your common sense. If you know the mechanism behind it, it will be easy for you to retain. Yes or no? So that is the thing. That is the thing. So antibiotic is of little value in cases of carrier. In cases, In carrier, you will give only antibiotic, right? Isolation period is like COVID only. So either for 14 days, like COVID, the isolation period is there because the patient is infective now. By coughing and sneezing, he is spreading everywhere. So isolate the person, isolate the patient at least for 14 days. Or until 2 consecutive nose swabs or throat swabs are negative. 2 consecutive swabs should be negative. It should be reported as negative. So whatever is early, या तो 14 दिन तक isolate करो, या तो उससे पहले relieve करना है, तो do twice. 24 hour apart, दो swab करो, double confirm करो that it is negative, then you can release it. So whatever is earlier, you can do that. So that is the isolation period, same as that of COVID. You got my point? You got my point? Coming on prophylaxis. So in COVID also, we were worried about the prophylaxis now. So someone in the family have COVID, we call the doctors. In my family, my mother, my father, my sister, my friend is infected, my roommate is infected, but I am not having infection till now. So can you give me some prophylaxis? Prophylaxis is something taken before infection. So prophylaxis ke liye kya? Diphtheria is also spreading like droplet infection now. So here carriers are 95 and cases are only 5. So that's why prophylaxis is important. 2 type of prophylaxis are there. One, prophylaxis given to the close contact. So close contact mein aap pehle hi antibiotic de do na. Before bacteria come in them. Just suppose my roommate, my friend, my any person in close contact to me having diphtheria, it usually occurs in children. So some child in the family have diphtheria, and so all the family members को आप पहले ही erythromycin or penicillin दे दो. So if the bacteria enters, already the blood have erythromycin or penicillin that can kill the bacteria. So that can be prophylaxis for the close contact. Give me a thumbs up. Give me a thumbs up. Yes or no? Yes or no? So give antibiotic, give diphtheria antitoxin previously only, and give vaccination. The vaccination is DPT. I guess everyone is vaccinated for DPT. Are you vaccinated for DPT? Yes, our parents have vaccinated us by DPT when we were kid, when we were child. But many of the person from the villages, from the periphery, they don't know about the vaccine. The children, they are not vaccinated for DPT. If they are vaccinated for DPT, nothing has to be done. Only a booster dose will suffice. If vaccination is not done, give proper vaccination of the DPT. So for prophylaxis, do three things. Give antibiotic, give antitoxin, and give vaccination if not previously vaccinated. If previously vaccinated, give only booster. So these are the 3 things you have to go for prophylaxis and close contact. So someone, imagine a family, the child is having diphtheria, diagnosed as diphtheria. Mother cannot be separated with the child. The child is small, 2 year, 3 year, 1 year. We cannot ask the mother to isolate the child alone in a room and don't go in the room. No one go. No, it is not possible. So of course, mother or the caretaker will take care of the child. So what about the mother? What you will give to the mother? To the mother, we will give 3 things. Antibiotic, that is erythromycin or penicillin. We will give antitoxin also, and we will ask the mother, are you vaccinated for DPT? Are you sure that your parents have vaccinated you DPT? If you are not sure, give the complete course of DPT. If you are vaccinated, take a booster. So that's it. That is prophylaxis to the close contact. Give me a thumbs up. Give me a thumbs up. For prophylaxis in the community, we already gave the vaccine DPT. You know about DPT. DPT mein there are 3 vaccines: diphtheria, pertussis, and tetanus. So it comes as a conjugate vaccine. We are not giving vaccine only for diphtheria. We are giving vaccine for the 3 things: diphtheria, pertussis, and tetanus. So this is national immunization. This is national immunization protocol. National. So DPT given 1, 2, 3. 3 doses of DPT, then booster of DPT. The 2 booster, total 5 doses of DPT are given in the entire life, right? So first dose of DPT is given at 6 weeks of life when the child is 1.5 months old. The second dose is given at 10 weeks. The third dose is given at 14 weeks. I guess everyone knows it. National immunization schedule, I guess everyone knows in the PSM. Yes or no? So at 6 weeks, 10 weeks, and 14 weeks, we are giving DPT-1, DPT-2, and DPT-3. DPT booster is given. The first booster is given at 24 months. And the second booster is given at 5 to 6 years. So 5 to 6 years pe last DPT is given. So although we all are vaccinated for DPT, our parents have vaccinated for the DPT. Give me a thumbs up. So this is the national vaccination, national immunization schedule. We all got 3 DPT, 1, 2, 3, and 2 boosters. So total 5 DPT is given. The first dose is at the 6th week, right? So DPT comes in various combinations, right? Usually it is a triple vaccine. Triple vaccine mein there are 3 things. DPT, diphtheria, tetanus, and toxoid. DPT में the D is actually toxoid, the inactivated toxin of the bacteria. Tetanus maybe it is toxoid, the inactive toxin of the bacteria. But pertussis is not toxoid, it is real bacteria which are killed. Real bacteria, real bacilli which are heat killed. It is the killed bacteria. Give me a thumbs up. So these two are toxoid, but this one, the middle one, that is P, is not toxoid. It is a bacteria. That is the conjugated vaccine DPT. I guess everyone knows it. So D and T are the toxoid, but the pertussis is the killed bacteria. It is not a toxoid. And pertussis increases the potency of other two. Okay. In DPT, pertussis increases the potency of other two. If without pertussis you give only D or you give only T, it is not that much potent. So it increases the potency of other two, pertussis. The dose is 0.5 ml. It is always given intramuscular on the thigh, the middle 1/3 of the thigh. Can you see the first DPT? Have you attended your pediatrics postings? If you have attended your pediatrics wards or posting, you maybe know how to give DPT. The first DPT given at 6 weeks, it is given intramuscular. It is given in the thigh, the middle 1/3 of the thigh. So in the thigh, the middle 1/3 of the thigh, on the lateral side, the doctor gives, the pediatrician gives the DPT, right? That is the DPT. DPT दो brand के आते हैं. Now in COVID vaccine we are having multiple brands. Now you may be knowing. So you are vaccinated by which brand? So COVID Shield is there. There are various brands. So for DPT also two brands are available in the market. One is Glaxo, one is Kasoli. So a little bit difference in the composition is there. D, P and T. See D, it is 25 Loeffler here and 30 Loeffler here. See T, it is 5 Loeffler here and 10 Loeffler here. And pertussis, it is 20 million and 32 million, right? Along with it, we add aluminium phosphate and thiomersol. What is the role of other two? Aluminium phosphate is acting as an adjuvant. It increases the immunogenicity of the vaccine, and thimerosal is a preservative. So total 5 things we are giving: D, P, T, aluminium phosphate and thimerosal. Aluminium phosphate is adjuvant and thimerosal is preservative. And DPT is the actual vaccine. You should know the doses according to Glaxo, according to Kasoli. Everyone give me a thumbs up. That's all about DPT. So the vaccine should be kept in the cold storage. 2 to 8 degree, it should be kept in fridge, not freezer. It should be like COVID, it should be kept in fridge, right? And once it is frozen, it accidentally agar aapne freezer mein rakh diya upar, so you have to discard it. You have to discard it. No exposure to the sunlight. Pack it in cartons, right? And once you open a vial, like in COVID also, now in one vial there were 10 persons we were giving. So one vial contained 10 ml. We are giving 1 ml to each person. So when 10 persons were accumulated, then only we open a vial. You may have attended your posting for COVID vaccines now. So like this only, there are 10 persons or many persons, many child from one vial. If something is remaining, discard it. Just discard it. Don't use it for the next session, right? That is the thing. So there are 2 contraindications for DPT vaccine. One is hypersensitivity, one is neurological disorder. That's it, right? After DPT, the child have fever. So warn the mother, warn the parents that I'm a pediatrician, I'm giving DPT to your child, I'm a doctor, and after that your child may have fever. So don't get panic, just give paracetamol or antipyretic to the child as and when required, SOS basis. So that is the fever. So take home remedy basis, don't admit the child for that, right? Immunization cannot prevent the carrier stage. In immunization, basically you are giving antitoxin. Listen my word. Let me draw the same diagram again. So these are the 2 tonsil. These are the blood vessel and these are the organs. This is kidney, this is heart, and these are the nerves. Tell me the complete story. What about the cases? In cases, the bacteria is present in the tonsil, produce the toxin here, and toxin, after producing toxin, the toxin enters the blood. And after entering blood, the toxin damages the organ. In vaccine, what you are giving? In vaccine, we are giving toxoid. Toxoid will cause inactivation of the toxin. Inactivation of the toxin, not the bacteria. So toxoid is the vaccine, or the toxoid can prevent the cases. But in carrier, only bacteria is coming. Toxin is not produced. So toxoid, if you are vaccinated for DPT, I can be sure that I will not be a case for diphtheria. But I am not sure whether I am carrier or not. I can be carrier. So vaccine do not prevent carrier state, because vaccine contain toxoid, and toxoid cannot prevent bacteria. Toxoid can prevent toxin production, but it cannot prevent the bacteria. If some bacteria is coming in me and residing inside my nose or throat, I cannot, oppose them, because my blood contains toxoid. It do not contain antibiotic, right? So vaccine prevent immunization, or vaccine prevent the cases. It cannot prevent the carrier. Carrier के लिए कोई vaccine नहीं है. If you are vaccinated for DPT, you cannot be a case, but you can be a carrier. You can be a carrier. Give me a thumbs up if you got my point. Give me a thumbs up if you got my point. Yes. DPT ka route you are asking. So DPT ka route is intramuscular. Middle 1/3 thigh, it is intramuscular. right? So that is about the immunization. I guess I'm completely done with Corynebacterium diphtheria. Let me revise the complete chapter once at a glance, and let me come on the next chapter immediately, Bacillus anthrax. Everyone give me a thumbs up first. Everyone give me a thumbs up first. Everyone give me a thumbs up. Shall I start? Shall I start? So let me, please help me in revising the entire chapter. Can you help me in revising? In the introduction, I have told you the name of the 4 scientists. The scientist who has discovered the disease, the scientist who has discovered the bacteria, the scientist who has discovered the toxin, the scientist who has discovered the antitoxin. If you have missed, watch the recording. The scientist who has discovered the bacteria is Klebsiella. That's why it is known as Klebsiella bacilli. For other scientists, you learn the names by yourself. Classification, it is of 3 types: gravis, intermedius, mitis, based on the severity. Gravis causes severe disease, intermedius causes intermediate disease, and mitis causes mild disease. Coming on the morphology. Who will tell me the morphology? These are gram-positive bacilli which occurs in the form of angular fashion because incomplete separation of the bacilli, and it is looking like a Chinese letter arrangement. At the ends, it is having metachromatic granules. You know everything about the metachromatic granules. These are non-capsulated, non-spore forming, non-motile. You know the morphology. Coming on culture. Coming on culture. Who will tell me the summary of the culture? In the culture, okay, just a minute. Okay. No, again, I will zoom it. Okay. Tell me 3 culture. Okay, just a second. Tell me 4 culture characteristics. 1, 2, 3, 4. And tell me 4 biochemical properties. 1, 2, 3, 4. That's it. Who will tell me? Someone among the audience, Priya, Nisha, King of Russia, anyone, Assad, anyone. Kindly tell me culture. Culture means the first culture is the liquid culture. What is the name of the liquid culture? His water serum. His water serum, right? That is the liquid culture. Solid culture is Loeffler serum slope. Loeffler serum slope. There are 2 selective medium. The first is telluride medium and second is Tinsdale medium. Both of them are blood agar. Telluride contains only potassium telluride in the blood agar. Tinsdale contains potassium telluride along with cysteine in the blood agar. These 2 are selective medium. Tell me the summary of all 4. Who will tell me the summary of all 4 one by one? In his water serum, it is turbidity. Very easy. In Loeffler serum slope, cream glistering colonies. In telluride medium, black colonies. Black color colonies. Based on that, the three you already know: gravis, intermedius, and mitis. In gravis, it is JC head colony. In intermedius, it is frog egg colony. In mitis, it is poached egg colony. I have shown you the diagrams. Those who have missed, please watch the recording. Beautiful diagrams I have shown here. Based on that, we are dividing the Corynebacterium diphtheria into 3 classes, 3 categories. Tinsdale medium, again black colonies with a brown halo. Everyone give me a thumbs up. That is the summary of the culture. We are done with culture. Now, can you tell me 4 biochemical reaction? The first biochemical reaction is fermentation of sugar. The second is they are H2S positive. The third, they convert nitrate to nitrite. And fourth, they are PYR positive. In the sugars, they ferment only glucose and maltose, no other sugar. H2S positive, nitrate to nitrate, and PYR positive, that you have to learn. We are done with culture also, biochemical properties also, morpho also, classification also, introduction also. Antigenic structure not important, virulence, resistance not important. Directly coming on the virulence factor. Everyone give me a thumbs up. Shall I come on the virulence factor? Who will give me the summary of the virulence factor? Virulence factor is only one toxin. The toxin produced by the bacteria, the diphtheria toxin. The toxin produced by the bacteria, the diphtheria toxin. It is having 2 fragments, fragment A and B. Both are important for toxigenicity. It is very lethal. Toxin can be converted to toxoid by 3 mechanisms: heat, formalin, and acidic pH. Right? And iron is required for toxin production. We can convert toxigenic, non-toxigenic strain to toxigenic strain. We can convert with the help of a beta phage, and the mechanism is known as lysogenic conversion. I taught you everything about the toxin. And what is the mechanism of action of toxin? It inhibits protein synthesis in the body. By inhibiting protein synthesis, it is acting. It acts on 3 organs. So basically, it acts on the adrenal, the heart, and the nerves. So 3 organs. The toxin damages 3 organs in human body. That is all about virulence factor. Coming on pathogenesis. The next heading is pathogenesis. Let me erase it and then again zoom it. So coming on pathogenesis. Who will tell me pathogenesis? In pathogenesis, it causes only one disease. The name of the disease is diphtheria. Diphtheria is having 3 effects: local effect, systemic effect, and complication. You already know local effect में pseudomembrane is formed. Systemic effect में the 3 organs are damaged. That is the heart, that is nerves and adrenal. Local effects are produced by bacteria. Systemic effects are produced by toxin. And complications can be by pseudomembrane also, mechanical obstruction, and heart failure can be there. Nerves में paralysis can be there. Most important, ocular nerve is involved. Lab diagnosis, you already know the most important test in lab diagnosis is Elix gel precipitation test. I have taught you very clearly. Coming on the treatment. Treatment means treatment of cases and treatment of the carrier. In the cases, you will give antibiotic as well as antitoxin. But in carrier, you will give only antitoxin. Right? Prophylaxis ke liye, we are having diphtheria vaccine. I told you everything about the diphtheria vaccine, that is DPT vaccine. So what is the dose? What is the route? What is the schedule? You know everything. So that is the summary of Corynebacterium diphtheria. In this way, you should complete Corynebacterium diphtheria. Let me come on the next bacteria, Bacillus anthrax. Are you people there? Shall I start the next bacteria? Shall I start the next bacteria? Bacillus anthrax. I'm starting the next bacteria, gram-positive bacilli, Bacillus anthrax. Everyone give me a thumbs up. Shall I start Bacillus anthrax? Yes. So Bacillus anthrax is also a gram-positive bacteria like Corynebacterium diphtheria. Only difference, it produces spore. Corynebacterium diphtheria was not producing spore. It produces spore. Right? So spores are produced by two bacteria. Aerobic में Bacillus है और anaerobic में Clostridium. Right? So I'm teaching you spore forming bacteria. You already know the mnemonic, BSc chemistry. In the BSc chemistry, B is Bacillus. Bacillus anthrax. It produces spores. Right? There are two Bacillus. In the classification, write down two names. Bacillus anthrax and Bacillus cereus. Bacillus anthrax causes the disease anthrax. And Bacillus cereus causes food poisoning. The two Bacillus are in your syllabus. Give me a thumbs up. I'm starting with Bacillus anthrax. The main Bacillus is anthrax. I will tell you cereus also in few lines in the end. Let me first teach you Bacillus anthrax. Give me a thumbs up. In the introduction, write down it is category A bioterrorism. You know category A, high priority bioterrorism. And men get infected secondarily. Basically, it is a disease of animals. So Bacillus anthrax is not a disease of man. Basically, it is a disease of animals, man infected secondarily. Bacillus anthrax. In the history, you can write down the name of the scientist if you wish. Questions can come. Usually MCQs come. So actually, it is the first bacteria which is seen under microscope by a scientist, Polander. It is the first bacteria which is transmitted by infected blood from one person to another person. It was discovered by Devine, the scientist Devine. It is the bacteria which is first bacteria which is isolated in pure culture by scientist Robert Koch, and he got Nobel Prize for that, right? And it is the first bacteria for which vaccine is produced by Louis Pasteur. So there are four scientists. Polander, that is the first scientist who has seen the bacteria under microscope. Devine, that is the scientist who has proven that that can be transmitted by infected blood. Robert Koch has isolated that, and Louis Pasteur have created vaccine for that. So write the name of the four scientists here. The four scientists. I have told you four scientists here also, four scientists here also. Everyone give me a thumbs up. Scientists are important for MCQs, especially in FMG exam. Give me a thumbs up. Coming on the classification, I already told you the classification, Bacillus anthrax and Bacillus cereus. Coming on morphology now. In the morphology, these are gram-positive bacillus, right? You can see the gram-positive bacillus, they are present in the form of the chains, one behind the other like a rod. One behind the other, they are present. So see, this is first bacteria, this is second bacteria, this is third bacteria, this is fourth bacteria. Likewise, they are present. Inside each of them, spore is present. Inside each of them. So spores are present, but no capsule, no motility. Spores are present. These are non-motile. These are spore forming, and usually they are non-capsulated, but some stents are capsulated, right? You know the mnemonic. Okay, I'm sorry. It is also capsulated. See the mnemonic of capsulated. You know the mnemonic for capsulated bacteria. I taught you this many times. PACIB MCV का जो B है, that is Bacillus anthrax. So capsule is present. See the mnemonic for spore forming. Spore forming का mnemonic है BSc chemistry. BSc chemistry का जो B है, it is Bacillus anthrax. But see the mnemonic for motility. Cute baby sleep and very protective solution HCl. Bacillus anthrax is not coming here, so it is non-motile. What is the summary? What is the summary? The summary is that capsule present, spore present, motility absent, and it is gram-positive bacilli. This is the summary of the morphology in front of you. Give me a thumbs up. What's the summary of the morphology? It is gram-positive bacilli, capsule is present, spore is present, but motility is absent. Right? I will give you a few points on capsule and spore. Capsule present, but capsule is not polysaccharide. Usually polysaccharide capsule है. यहाँ पे capsule is polypeptide. If it is polypeptide, name the amino acid जिसका polymer है. So amino acid is D-glutamic acid. So it is a polymer of D-glutamic acid. Capsule is polypeptide, not polysaccharide. That is the main point in the capsule. Spores are oval and central. Spores are oval. Can you see the spores are oval and they are central. Inside each bacteria, this is a bacilli. The spore is oval and central. The spore is oval and central. So spores are oval and central and they do not cause bulge. They do not cause the bulge. You got my point? That is about the spores. Now, do a special stain, Giemsa stain. If we do Giemsa stain, the capsule convert red in color and the bacteria convert purple in color. You see the red and purple color. Can you see the red color here is the capsule, the degenerated capsule. Capsule got degenerated. And can you see the purple color? The purple color is the bacteria. Purple is the bacteria. So on Giemsa stain, the bacteria become purple and the capsule become red. And this is known as okay, that is on Giemsa stain. You learn the colors. There is another stain which is known as polychrome methylene blue. On polychrome methylene blue, the degenerated capsule can be seen in the form of the purple clusters. You see, these are the purple clusters. That is the degenerated capsule. It is known as Mac-Faridian reaction. On polychromatic methylene blue, Mac-Faridian reaction. इस पे question आता है. What is Mac-Faridian reaction? So on polychromatic methylene blue, the purple color amorphous material is the capsule which is disintegrated from the bacteria. And this is known as Mac-Faridian reaction. Please learn. It is polychromatic methylene blue. On polychromatic methylene blue, Mac-Faridian reaction is seen in which the capsule become degenerated and it looks purple color. This is the summary. You got my point? You got my point? And bacteria arranged one behind the other in the form of the bamboo stick. One behind the other in the form of the bamboo stick. This is known as bamboo stick appearance. Bamboo stick appearance. So what I have taught you till now, let me summarize and then I will move on the culture and biochemistry. I have told you the introduction, the name of the 4 scientists, right? In the classification, only 2 bacteria are there, Bacillus anthrax and Bacillus cereus. You know the 2 of them, I will describe both of them. In the morphology, these are gram-positive bacilli, capsule present, spore present, but motility absent. The capsule is a polypeptide. It is not polysaccharide. Polypeptide made up of D-glutamic acid. Spores are present which are central and oval and non-bulging. And motility is absent. Right, that is the summary. Coming on culture. Everyone give me a thumbs up. I am coming on the culture now. In the culture, I will tell you 5 cultures. Write one name in front of each of them. On nutrient agar, write down Medusa head appearance or frosted glass appearance. What do you mean by Medusa? Imagine Medusa is a girl. Medusa is a girl who is having curly hairs. Curly hairs. Can you see the Medusa head appearance? The curly hair. The curly hair is due to the capsule. Can you see? This appearance on nutrient agar is known as Medusa head. So this is the meaning of Medusa. Medusa is a girl having a curly hair. So this appearance is known as Medusa head appearance. Right? Number 1. You see, this is again Medusa head. Medusa head means curly appearance. Medusa head appearance. So this is seen on nutrient agar. On nutrient agar. Give me a thumbs up. You see the frosted glass. It is translucent, not transparent. The color of the colony is translucent. Can you see the color of the colony on nutrient agar? The color of the colony is translucent. That's why it is known as frosted glass appearance. Frosted glass. You got my point? Now the 2 appearance on nutrient agar. What is the summary? Medusa head and frosted glass. On nutrient agar, either Medusa head or frosted agar. Medusa head is the curly hair. That is the mated hair of a girl Medusa. That is long interlacing chains of the bacilli are there. Nutrient agar. Right? And frosted appearance, frosted glass. On the blood agar, it is gamma, alpha, beta, gamma. It is gamma hemolysis. No hemolysis. That is gamma. On gelatin swab, it is inverted for appearance. You know, okay, let me take a gelatin. In a test tube, we will take gelatin. This is gelatin. It is filled till here. If I'm putting Bacillus anthrax here, so it will start liquefying the gelatin. So liquefaction is like this. You know, it is a fir tree. Have you seen the Christmas tree? Christmas tree or a fir tree? The liquefaction is in this shape. It is looking like an inverted tree. It is known as inverted fir appearance. Give me a thumbs up. Inverted Christmas tree appearance. Please appreciate the inverted fir appearance on the gelatin. Give me a thumbs up. This is known as inverted fir appearance because maximum oxygen is present at the top. Maximum liquefaction is at the top. And as we go down, the oxygen decreases. So liquefaction decreases. So gelatin is maximum liquefied at the top because of the aerobic condition. So it is inverted fir appearance. Give me a thumbs up. Inverted fir appearance. If you can zoom out and see this image, it is inverted fir. So maximum liquefaction at the top because of the oxygen or aerobic condition. Give me a thumbs up. Everyone give me a thumbs up. This is inverted for appearance. Selective media of Bacillus anthrax, very important, PLET media. What is the full form of PLET if you want to learn? P stands for polymyxin, L for lysozyme, E for EDTA, and T for thallose acetate. So PLET medium. These four things kill all other bacteria except Bacillus anthrax. So in these four chemicals, only Bacillus anthrax will survive, rest all will die. So that is the selective media. You got my point. In PLET medium, that is the selective medium, you have to learn the name. The last one is the solid medium containing penicillin. Solid medium containing penicillin, you see the colonies. The colony is looking like a pearls. You know pearls यानी मोती. Pearls. The string of pearls. मोतियों के हार जैसा लग रहा है. String of pearls. It is looking like string of pearls. Give me a thumbs up. It is looking like string of pearls. Can you tell me the summary of the 5? Who will tell me the summary? Culture media is done. The summary is in front of you. Who will tell me the summary of culture media? On nutrient agar, write down medusa head appearance or frosted glass appearance. Frosted glass appearance. On blood agar, write down gamma hemolysis, no hemolysis. On gelatin swab, write down inverted fur appearance. The selective media, write down the name, the full form PLET. You know the full form of PLET. Polymyxin, lysozyme, EDTA, and thallose acetate. Right. Solid media containing penicillin, string of pearl appearance. String of pearl appearance. Everyone give me a thumbs up. इस पे बहुत MCQs भी आते हैं and they are important for theory exam also. Give me a thumbs up. So I'm done with the 5 culture media. You already know the summary of them. Coming on biochemical reaction. Fermentation of sugar, they ferment 3 sugar: glucose, maltose, sucrose. Glucose plus maltose plus sucrose with acid, no gas. That is, they are catalase positive. They reduce nitrate to nitrite. Only 3 biochemical reactions are important. In antigenic property, capsule is there. Capsule is made up of polypeptide, not polysaccharide, which is a polymer of D-glutamic acid. Right? And somatic antigens are there. Okay. It produces spores. So, spores, it is basically disease of animals. So it is basically present in the sheep, in the cow, in the skin of the sheep, skin of the cow. From the sheep and cow, the woolens are made. So whenever we separate the fur, the skin of the sheep, cow, or other animals, they can contain the spores of Bacillus anthrax if the animal is having a disease of anthrax. So before making woolens or any leather material or anything out of the animal products, we should kill the spores. So killing of spores is known as duckering. Duckering is destruction of the spore in the animal product. Animal product can be, the skin of the cow, the skin of any other animal. So duckering is done by treating the animal product with formaldehyde. Once we treat it with formaldehyde or we heat it at 60 degrees for 6 hours, the spores are killed. You see, this is the animal product, inside which spores can be present if the animal have infection. Once we convert the animal product into woolens, leather, purse, shoes, and the spores can be present and they can infect the human. So before making any product out of the animal material, we have to kill the spores, and killing the spores is known as duckering. Duckering is destruction of spores done either by heating or by treating with formaldehyde. You got my point. So we are done with antigenic structure also. Coming on virulence. Virulence is of 2 types: toxin and capsule. It produces toxin. Toxin is having 3 factors: edema factor, factor 1, protective antigen, factor 2, and lethal factor, factor 3. So that is the toxin. Toxin basically inhibit ATP formation. It causes accumulation of cyclic AMP. In the cell, cyclic AMP increases, that causes cell death. So okay, basically let me draw a human cell. This is a cell. This is the host cell. Host cell can be human, can be animal. Now this is the toxin of Bacillus anthrax. This is the toxin of Bacillus anthrax, having 3 factors: edema factor, protective antigen, and lethal factor. 1, 2, 3, 3 factors are there. The toxin will enter inside the cell, and after entering inside the cell, it will stimulate an enzyme. The name of the enzyme is adenyl cyclase. Adenyl cyclase convert ATP to cyclic AMP. More and more cyclic AMP is formed. This cyclic AMP, it will activate the sodium channels present on the surface. So more and more sodium will enter inside the cell. Along with sodium, more and more water enter inside the cell. The cell will swell, the cell will swell, the cell will burst. The cell will burst, the cell will die. The cell will be dead. Give me a thumbs up. This is the mechanism how toxin act. So basically toxin increases cyclic AMP concentration inside the cell. Can you imagine the diphtheria toxin in the last chapter I taught you? Diphtheria toxin inhibit protein synthesis. But here Bacillus anthrax toxin increases cyclic AMP concentration, because of which it is killing the cell. Right? Give me a thumbs up. Capsule, you already know the capsule is polypeptide, not polysaccharide. Polypeptide with the amino acid is glutamate. It is polyglutamate. I am done with virulence also. Coming on pathogenesis. Pathogenesis. Coming on pathogenesis. It causes a disease known as anthrax. Only one disease, anthrax. Anthrax is basically a disease of animals like cattle and sheep. Man is the accidental host. It occurs in herbivores. It is more common in herbivores than carnivores. It is a zoonosis. Man is the accidental host, right? So how it is coming to the man? How it is coming? It comes secondarily to the man from that diseased animal. There are three routes. Either you come in contact with the infected animal or the product of the infected animal. Just suppose I am having a leather purse or I am wearing some woolen. So that woolen or the leather purse is made up of from the skin of an animal that contains the spores. So I will be infected. So that is contact with the infected animal or the product of the infected animal. It can be insect bite. There is an insect who is biting the animal and the same insect coming and sitting on me and biting me. It can be ingestion. If someone is ingesting the animal who is infected with the anthrax, and inhalation. So there is an animal, there is a butcher place where the spores are present in the air. So via inhalation also it can be there. Man-to-man transmission is not there. That is the rules. Now anthrax is of three types: cutaneous, pulmonary, and intestinal. The three type of anthrax. Cutaneous is known as Hyde-Potter disease, right? Here, the patient have a pustule at the point of the entry. As pustule, papule is there, it will convert into pustule, and then it will convert into black issue known as malignant pustule. So first, papule convert into pustule, and then malignant pustule is formed. So that is skin, that is cutaneous, right? And ultimately, it can enter in the blood, and from blood, it can go to the meninges, causing meningitis. That is cutaneous one. The second is the pulmonary. It is known as Wool-Sorter disease. In the lung, it will cause pneumonia. It will cause bronchopneumonia or pneumonia. Third is intestinal. It occurs by eating the animal, meat of the animal who is infected with anthrax. So the patient will have diarrhea. Patient will have vomiting and diarrhea. That is the three type of the anthrax in front of you. It can be cutaneous, it can be pulmonary, it can be GIT. Cutaneous have papule and pustule. Pulmonary have pneumonia, and GIT have vomiting and diarrhea. This is the summary. Give me a thumbs up. The three type of anthrax. I'm done with pathogenesis. Lab diagnosis you can do by yourself. Not very important. We have already done Mac-Faridian reaction. We have done the 5 type of culture. Nutrient agar pe medusa head colony. Blood agar pe gamma hemolysis. Gelatin pe inverted fur appearance. Selective media is PLET. And solid media containing penicillin pe string of pearls. By all these mechanisms, we can do the lab diagnosis. Treatment and profile. Prophylaxis के लिए no vaccine is available. Vaccine is available for the animals, not for humans. And treatment is doxycycline and ciprofloxacin. It is the drug of choice. Animal vaccine is available. I am done. I am done with Corynebacterium diphtheria and Bacillus anthrax. Do you have any doubt till now? Till now, from the morning, I have covered Staph aureus. I have covered Streptococcus pyogenes. I have covered Pneumococcus. I have covered Neisseria meningitis, meningococcus. I have covered Neisseria gonorrhoeae, gonococcus. I have covered Corynebacterium diphtheria. I have covered diphtheroids. So basically, I have covered gram-positive cocci. I have covered gram-negative cocci. I have covered gram-positive bacilli. Still gram-negative bacilli are remaining. So I will arrange some other day, maybe tomorrow or a day after tomorrow, another lecture, that is episode number 4 of bacteriology, in which basically I will cover gram-negative bacilli. In gram-negative bacilli, important one are E. coli, Klebsiella, Proteus, Salmonella, Shigella, and Vibrio. These are the important ones that I have to do in a comparative analysis, same as I'm doing here. That is gram-negative bacilli. So I will arrange some other lecture. Currently, I would like to stop here. Thank you very much for being with me. Thank you very much for giving your precious time to me. And there are few announcements for you. Let me do those announcements. Just a second. Okay. So, that my next lecture will be tomorrow. I will drop the links on the Telegram and the WhatsApp. If you are taking any subscription of Unacademy today, that is till 31st May, you will get it at the same price. From tomorrow, price hike is there. 20% price hike is there. Right? If you are preparing for NEET PG 2023 and taking a subscription for 6 months, you will get additional 2 months at the same price. These are the new batches we have launched for the preparation. Academy have launched UPSC CMS preparation also, right? So you can take the subscription if you're preparing for the UPSC. If you want subscription only for Prof 1, that is anatomy, biochemistry and physiology, you can take subscription of Prof 1 only. These are the various subscription plans available with us. In the Plus, these are the plan. The Iconic, these are the plan. In the Light subscription, these are the plan. MBBS Prof 1 and UPSC. See the prices, see the plans, and if you apply my code Sachdev10 on any of them, you will get straightforward 10% discount right now. Thank you very much for being with me. Bye bye. Study hard. All the best. Good night. And I'm ending this lecture.